Hi class, this is lecture 3, large biological molecules. So we started last week talking about kind of the fundamentals of chemistry, the basics of chemistry, and so this lecture we talk about these important molecules that are important to our cells and our bodies, these important biological molecules. So the 4 important classes of biological molecules are carbohydrates, proteins, nucleic acids, and lipids. And so these are all, well, 3 of these are classes of polymers. And so a polymer is like a really long, large molecule made of smaller building blocks called monomers. And so you could kind of see how carbohydrates is a polymer of these little rings. And so similarly for these other 2. So carbohydrates, proteins, and nucleic acids are polymers made of monomers. And lipids are not technically true polymers, but they are an important class of biological molecules, so we'll talk about them today. So that's kind of like the preview. So large polymers are known as macromolecules for their huge size. And so it's not like small, simple molecules like carbon dioxide or oxygen where it's just a few atoms. These molecules are large, and so they're called macromolecules. And so a polymer, again, is a long molecule consisting of many similar building blocks. And then there can be different kinds of polymers, but you know, that category is made of that kind of building block. And so the repeating units that serve as the building blocks are called monomers. And so here is an example of just, you know, like a block. And so carbohydrates, proteins, and nucleic acids are polymers. They're each a different class of polymers, but they're, you know, combined in that they're polymers made of monomers. So how do we make these huge macromolecules? So enzymes are specialized macromolecules that help speed up chemical reactions like the ones that make or break down polymers. So to join 2 monomers together to build a polymer, you have to kind of connect one monomer to the next one. And so a reaction called a dehydration reaction occurs when those 2 monomers bond together through the loss of a water molecule. So let me move forward one slide just to show you that. So on the top, panel A is a dehydration reaction when you're building a polymer. So if you see the word synthesizing, it's making or building. So when you're building a polymer, you do a dehydration reaction. So on this hand, on this left part is a short polymer. And then to the right is one unlinked monomer. And so you're trying to add on to this already short polymer so you could build it even bigger. And so the hydrogen coming off the end of this molecule, and then the hydrogen and oxygen coming off of this molecule leave together, so they rearrange dance partners like chemical reactions, and so they leave all together as a water molecule. So because the water molecule leaves, this is why it's called like dehydration reaction, because the water molecule gets removed. And so that's how you link up one monomer to the next one. And that's a dehydration reaction. And then cells will also break down polymers so that they can use these pieces to maybe reform new monomers or be used for some kind of other molecule. So polymers are disassembled back into monomers through a reaction called hydrolysis. So hydrolysis is a reaction that's pretty much the reverse. And if you look at the word hydrolysis, hydro means water, typically as a prefix, and then lysis usually means to break. And that is totally relevant here. So here, if we go back to the picture, hydrolysis is breaking down a polymer. So if you were to add a water molecule back in, it rearranges, and then now it breaks that bond between these two monomers, between the 3 and 4. And then now there is kind of an end to this short polymer with the H. And then now there's kind of the end to this monomer with the OH over here. So hydrolysis, by adding water, you break this bond. So that is how you make and break polymers. And so one of the activities, also just to come back to this, one of the activities that you have for homework is to kind of show how this links up to the big picture. So we're talking about these molecules, but it's relevant because they do seem familiar, right? Some of these names like carbohydrates, protein, this sounds really analogous to like our diet, and it really is. Like, we get these molecules through our diet. And so when we eat these things as nutrients, our bodies have to be able to break them down and be able to get energy or build or use them to build new molecules in our cells. So that's what that part of the homework is supposed to help demonstrate. Okay, so to go back to talking more detail about these guys. So cells have thousands of different macromolecules. And they're going to vary. The macromolecules will vary among the cells of an organism. So we have specialized cells in our bodies. And so even in one organism, we're going to have cells that have different amounts of protein or different amounts of carbohydrates or other things like that. So the macromolecules will vary among the cells of an organism. They'll vary more within a species, and they'll definitely vary even more between species. So our cells and what make them up are going to be, pretty different. But these small 4 classes of the big macromolecules are really can make very diverse things. So a huge variety of polymers can be built from this small set of monomers. So as we talk about these different kinds of classes of molecules, this is a study tip from the textbook. So try to, for each one, list their monomer and then draw 2 examples of that macromolecule and list, how are they similar to each other? Why are they, both part of that kind of macromolecule? And then what do they do? So that can kind of help this information stick. It really does help to be able to draw things out yourself and put these things in your own words. Okay, and so class 1 that we're going to talk about is carbohydrates. And so you'll notice that I try to put different headings to kind of help show how there's categories of things. So this big heading is going to be Roman numeral 1, carbohydrates. And then there will be A, and then another one. So hopefully that helps organize these concepts into like these big groups and then smaller and smaller groups. So carbohydrates, they include sugars and polymers of sugars. So carbohydrates are a polymer, and the monomers are the little building blocks that we call monosaccharides, and we also more commonly call them simple sugars. So the simplest carbohydrates are monosaccharides, and yeah, we can just call them simple sugars. And then if you link one sugar to the next, then you are building a polymer of those sugars. So carbohydrate macromolecules are polysaccharides. So if you look at the prefix mono, means 1. And then saccharine, C-A-C-C-H-A-R, usually means sugar. So there's even like a vocab word like saccharin that describes people or something that's like overly sweet. So just to show that that means sugar. So a monosaccharide is just one sugar, a simple sugar. If you build more than one, then that's called a polysaccharide. Poly means many, so many sugars. So the big carbohydrate macromolecules are polysaccharides, polymers made of many sugar building blocks. And so now this is like the subcategory, sugars. We'll just focus on that for now. So monosaccharides, again, simple sugars, have molecular formulas that are usually multiples of C, 2 hydrogens, and then an O. So, carbon, 2 hydrogens, and 1 oxygen. But if you have, it'll be multiples of this. So it could be this. This is, pretty simple. Or it could be glucose. And so see how glucose is a multiple of this, same ratio. For every 1 carbon, if there's 6, you multiply that by 2, there's 12 hydrogens. And then so there's 6 oxygens. So that follows it. And so glucose is the most common monosaccharide. And monosaccharides are classified by the location of one of the functional groups, the carbonyl group, and the number of carbons in the carbon skeleton. So we'll look at some pictures. So for example, if the trioses have 3 carbons in this sugar. Triose. And in general, if you hear the ending ose, it's a sign usually that this is like a sugar. So trioses, 3 carbon sugars. And then depending on, again, where that carbonyl group is, if it's like at this position, that gives it this kind of name. It's an aldose because it's at the end. If it's at this other position, then it's a ketose. If, these are examples of pentoses. There's 5 carbon sugars, and then pent means 5, so 1, 2, 3, 4, 5, and then over here. So even if there's really slight differences in molecules, that's enough for it to be named a different molecule. So glyceraldehyde and dihydroxyacetone, there's a small difference, and so that's why they're named different things. These are both pentoses, but they have different names because the carbonyl group is in a different position. So ribose, ribulose, it's a sugar. Glucose, galactose, fructose. So often, they'll end in oses if they're sugars. So if there are 6 carbons, then it's a hexose. That means 6. And so again, you can kind of see how it follows that ratio of 1 to 2 to 1. And so sugars, even though we often draw them as the linear skeletons to show, the carbons, in aqueous solutions, so a throwback already, meaning a watery solution, many sugars end up forming rings. So they'll kind of orient in this way, and then these groups will kind of rearrange so that it's in a ring in this kind of configuration. And then, as I kind of mentioned, in especially organic chemistry, if we draw shapes, it's kind of assumed that each point on a shape is a carbon. So here in this ring form, we're literally pointing out which one's carbons. But in this abbreviated ring structure below, you can see that there's still a hexagon, but there's not actually a C there. It's kind of just implied that this point is a carbon, this point is a carbon. And then we'll usually number the carbons. So this is carbon 1, 2, 3, 4, 5. And then there's a 6th one right here. And so this second bullet point is in bold for a good reason. This is like a big point, main idea. So monosaccharides, again, simple sugars, serve as a major fuel for cells and as raw material for building molecules. Because remember, we talked about how carbons, form the skeleton of or the backbone of a bunch of things. So monosaccharides will typically be used as a major fuel for cells or raw material for building molecules, because so much stuff is built off of carbon, right? The carbon skeletons. So when we get to the, in a future lecture and we talk about metabolism and cell metabolism, how cells get energy, monosaccharides are a major fuel for that, especially glucose. We're going to focus on cellular respiration and how glucose specifically gets broken down and we obtain energy from that. So a monosaccharide is just one simple sugar. A disaccharide, di means two. So a disaccharide is formed when a dehydration reaction joins two monosaccharides. And there's a covalent bond here between these two monosaccharides, but we have a specific term for this kind of covalent bond. It's called a glycosidic linkage. So I try to teach all these different word parts because that way, if you learn the word parts, when you learn new information, you can kind of tell, oh, okay, if it has this part, then that means this. So a glycosidic linkage is between sugars. And in general, like moving forward, if you see the prefix glyco, that usually means sugar. So that's a big, helpful way to remember. A glycosidic linkage is the one that's linking two sugars, two monosaccharides. So here's an example of a dehydration reaction between two monosaccharides to make a disaccharide. So in the top panel A, there's two glucose molecules, so they're two identical molecules in this example. And it's two glucoses that you put together, and through the dehydration reaction, you build the disaccharide maltose. And then in panel B below is another example of a different kind of disaccharide. So in this case, you're building sucrose, and to do that, you put together a glucose and a fructose. So glucose and then a fructose looks slightly different. And so again, you do a dehydration reaction to build this guy, and in this case, you're building sucrose, another disaccharide. So it's kind of a similar process to build even bigger ones, just a series of dehydration reactions. So polysaccharides are bigger polymers of sugars that has many sugars. Poly, again, means many. So the polymers of sugars are polysaccharides, and they have major roles in cells. They have roles in storage, and then also in structural roles. And so I'll talk about examples of each one. And so the architecture or the structure and the function of a polysaccharide are determined by its monomers, what sugars are there making up this huge thing, this polysaccharide. And then also by the positions of those glycosidic linkages. So the first example of a storage polysaccharide. So again, this is where those headings come in. So under polysaccharides, now there's subheading storage polysaccharides. And so the first example is starch. So starch is a storage polysaccharide of plants, and it consists of glucose monomers. And so plants make energy from the sunlight, and so they'll store this energy in the form of, a bunch of glucose molecules linked together to make this molecule called starch. And so, plants store surplus starch as little granules that are stored inside of chloroplasts and other plastids. So that might not make that much sense yet, but that's how plants store their excess energy. And so there are actually different forms of starch, but the simplest form of starch is called amylose. And just to revisit this point, so carbohydrates, again, when we kind of like think big picture in the diet, you know, sugars are a carbohydrate. And so I always say that, like, if we think about diabetics, so for example, my mom is diabetic, she, you know, shouldn't eat sugar or things, you know, with glucose. And so that's something that she watches in her diet, but she also has to watch out for other forms of carbohydrates because they're usually made of a bunch of glucose units. And so, you know, people with diabetes are usually advised to not eat starchy vegetables. And this is what that means. So vegetables are plants, and plants store energy and glucose in these starch molecules. And so you're not recommended to eat starchy vegetables like potatoes and carrots and things like that because they're larger molecules literally made up of glucose and sugars. So just to kind of, again, tie things into big picture. And I will literally show a picture on the next slide. So plants store their energy in the form of starch. Glycogen is the molecule that is the storage polysaccharide in animals. And so these are stored mainly in the liver cells and muscle cells. And then we can break these down and then release the glucose when the demand for sugar increases. So here are pictures of, on the top panel, starch, and then on the bottom panel, glycogen. And you could see all these little rings, and yeah, those are all glucose. It's glucose monomers. So starch is literally made of like thousands of glucose monomers linked together. And when they get broken back down into glucose, you know, it'll raise your blood sugar glucose level. And so that is the storage polysaccharide in plants. And then in animals, it's glycogen. It looks pretty similar, but it's different enough again. It has its own name. And just to point out, so yeah, starch is kind of like a category also. So amylose is the most simple starch, and so it's unbranched, it's pretty linear. And then this other one is actually a second kind of starch. So this is called amylopectin. It's long, and then there are some branches where like a branch of a bunch of glucoses divert. And then the main branch keeps going, but then there might be smaller branches. So those are two kinds of starch. And then glycogen is branched. Okay, so those were storage polysaccharides. Another category of polysaccharides are structural polysaccharides. So these help provide some kind of structure to something. So the first example is cellulose. Cellulose is a major component of the tough wall of plant cells. So we haven't covered cells just yet, but plant cells are kind of like square looking, if you kind of remember to high school biology. And that's because they have a cell wall. And so this cell wall is typically made of cellulose. So like starch, cellulose is a polymer of glucose, but those glycosidic linkages are different. And that's based on that there's two ring forms for glucose. There's the alpha form and then the beta form of glucose. And so here, this picture is showing on the top. In the middle is the linear structure for glucose. On the left is the alpha glucose, and then on the right is the beta glucose. And so the main difference is kind of this opposite positioning of the hydroxyl groups. So in the beta glucose, these two hydroxyl groups are on opposite sides, and in the alpha glucose, they're on the same side. So panel B and C is showing starch versus cellulose. So their structures are overall very similar. But look how like those OH groups are all on the same side for starch. And in cellulose, they're on opposite sides for each glucose. So again, even if like the chemical name and overall structure is very similar, the slightest different like positioning makes it a totally different molecule. So starch versus cellulose. So yeah, cellulose, because of that slight difference, it kind of provides more structure, and that's why it's like a structural polysaccharide, and helps provide structure in the cell walls of plants. Starch in the alpha configuration is largely like helical. So I think I'll also show that. I guess there's not really a picture for that. Okay, so starch, again, because it's the alpha configuration, it kind of exists more as like a helix. And then in cellulose molecules, which have that beta configuration, that makes it kind of straight and unbranched. Yeah, and then some hydroxyl groups on the monomers of cellulose can bond with the hydroxyls of other cellulose molecules, and so that is kind of how it helps lay so flat, because of how these molecules are oriented and structured. And so, again, these small differences in kind of like orientation and structure of these chemical groups matter. So enzymes that can digest and break down starch by breaking down alpha linkages are specialized just for alpha linkages, and they can't hydrolyze beta linkages in cellulose. So in us humans, like, we can't break down cellulose. So that's why the cellulose in human food passes through our digestive tract, and that's actually what we know as fiber or dietary fiber, insoluble fiber, because we don't have the enzymes that can break down those linkages in cellulose. Although we do have enzymes that can break down starch. So again, even though the molecules are super similar, chemistry really matters. So if we can't break down fiber and dietary fiber and cellulose, then it kind of just goes right through us, and then it helps like push out everything else through our digestive system, and that's why we say like it keeps us regular. Yeah, because it's really just kind of pushing everything out, because we can't digest it. We don't have enzymes that can break down that kind of linkages. And so some microbes have the enzymes to digest cellulose, so bacteria. And so many herbivores, like cows, to termites, have symbiotic relationships with these microbes because they can break down the cellulose, and then they can end up together benefiting from breaking that down. So for example, termites, even though they eat wood, and wood is a plant, and they have plant cell walls made of cellulose, the termites can't break down the cellulose, but they have microbes in their tiny guts that can break down the cellulose. And so that's how they will like work together and have a symbiotic relationship to end up breaking that down. Oh, and then, I should fix this number. So another example of a structural polysaccharide, so the second example, I gotta fix this, is called chitin. It does look like it would be pronounced chitin, but I guess it's called chitin. So chitin is found in the exoskeleton of arthropods. So for example, bugs. This kind of is what you hear crunching when you step on a bug. It's the chitin. And then it also provides structural support for the cell walls of fungi. So fungi also have cell walls, kind of similar to plants, but they're made of totally different stuff. In this case, it's chitin, not cellulose. And so here's an example of chitin, and it's embedded in proteins, and that helps form the exoskeleton of arthropods. Gross. Okay, so this is a big picture summary of carbohydrates. So the components, they're made of monosaccharide monomers. There are different kinds of carbohydrates like monosaccharides, disaccharides, and polysaccharides, and these are the examples of each one. And then the different functions of those different kinds. Then maybe I'll just make this video several parts because I feel like it'll get really long. Okay, so we'll just pause here.